Most of you have met me or know me already, but I'm John David Rhodes, and I'm the warden of Leckhampton. So I'm a film historian by training, so I'm the least likely person, apart from the office that I hold, to give this introduction. But I'm really pleased to welcome Richard Henderson back, of course, to Leckhampton. Richard, by his own account, was born into a normal working class family in the borders of Scotland. His father was a baker and his mother held a variety of odd jobs, and he's a product of Scottish state school education in which he excelled and which delivered him to Edinburgh University, where he undertook his undergraduate degree in physics. Richard came to Cambridge to do a PhD in the MRC Laboratory of Molecular Biology in 1966. And arriving unfamiliar with the collegiate system, Richard sought advice from Keith Moffat, his contemporary from Edinburgh, who eventually became professor of mathematical physics at the university from 1980 to 2002. And Moffat sent back a letter of advice, and a small bit of this I just want to read because it's so charming. He writes to Richard, Corpus Christi has a truly magnificent postgraduate hostel called Leckhampton, ultra smooth, full of interesting people and much sought after. No molecular biologist, but plenty of chemists. An enlightened attitude towards research students, recommended. It's a bit out of town, but the setting more than makes up for the comparative isolation. A gigantic garden with a swimming pool, a rose garden, Henry Moore statue, squash courts and playing fields, the lot. So that obviously, that did the job of convincing Richard, who then lived for 18 months in the George Thompson building, and then a bit later, after marrying and starting a family, in one of the flats in Cranmer Road. After completing his PhD, Richard went to Yale, where he spent 3 years, and then returned to the MRC LMB in 1973, where he's been ever since, working as a structural biologist, investigating the structure of unusual biological molecules. In the mid-1970s, he began collaborating with Nigel Unwin on electron microscopy, and in 1999, the two shared the Grigory Amanoff Award of the Royal Swedish Academy of Sciences. Richard was director of the MRC LMB from 1996 to 2006, whose expansion he guided, and it was also in those years that he became an honorary fellow of the college. In 2017, a date that I'm sure all of us here are well aware of, the Nobel Prize in Chemistry was awarded to Richard, along with Jacques Dubouchet and Joachim Frank, and I'll quote from the award, for developing cryo-electron microscopy for the high-resolution structure determination of biomolecules in solution. And I guess I was just thinking as well about my fitness or unfitness to introduce someone, a scientist of this distinction, but what is so wonderful about the work is just its emphasis on the visual and making visible the tiniest particles of life. And just also anecdotally, just by chance, I was speaking to one of our newest, most recently inducted fellows, Nirupa Desai, and her research on cancer just regularly makes use of the technology that Richard's research has made possible. So in 2018, we were fortunate enough to be able to celebrate Richard's Nobel with a feast in the old house, where we also toasted the installation of his portrait in the hall. And in that portrait, Richard appears painted against the backdrop of these gardens of Leckhampton House and the GTB, Richard's home when first he came down from Scotland to Cambridge and to Corpus and to this place. The college delights in its long affiliation with Richard and is hugely gratified by the enthusiasm with which he accepted the invitation to deliver this lecture. And I want to emphasize as well that this lecture is also this year's Ahmed lecture, which is endowed through the generosity of Haroon Ahmed, a former master of this college, a sometime warden of Leckhampton, and it's a joy and privilege to have Haroon and Anne and their family here with us today. So please join me in welcoming Richard Henderson, whose lecture today is entitled Using Electron Microscopy to Understand the Molecules of Life. Richard.
Thank you. Thanks a lot. Yeah, so it's a great pleasure and great fun to be back here after something like 56 years from the time that I first came. And I will tell you all about how wonderful electron cryomicroscopy is and how it's still in a sort of ascendant trend. But I thought I might start by, and a lot of people won't have been here in 1966, although one or two in the audience are. But I thought I'd just tell you one or two of the things that happened. And I think I do have to redo the slides. I think somehow or other we've lost contact with the talk somehow or other. Yeah, now I've got it. So I was just going to start. So when I came, I was always, keen on photography, and one of my activities in Edinburgh was the Edinburgh University Photographic Society. So I had a lot of, so I've scanned a few of them from 1966. So this was the old house, Leckhampton House, as it was then, and it was the most desirable place to live in, and I never managed to get there. I started in Selwyn Gardens for a year, and then we're still in contact with the people that I knew who were neighbours in Selwyn Gardens, and then I had 6 or 9 months in the George Thompson building before moving out to a flat shared with some other students, actually. But I did get the community spirit, meeting people, having known nobody that was in Corpus at that time. And I should say, actually, for John David, that there are 2 Keith Moffats in the world. And the one that's prominent in Cambridge in the Department in Applied Maths and Theoretical Physics, who I know well and is from Scotland, and we met for Burns Supper this year, actually. But there's another Keith Moffat, who was the one a year ahead of me, who wrote this letter. And he is now a professor in Chicago University. So, but there's probably, several thousand Keith Moffats in the world, so it's not too bad. The 2 other stories I thought I would tell you about Leckhampton was, in our first year, many of us who lived here, this was the furthest south I'd been in the whole world. At age 21 or something like that. So there were a few of us, 6 young men, because there were no women in the college at that point. We decided we needed to go on an expedition in the first summer. So that would be about 8 or 9 months after starting in Leckhampton. And we had 2 very old cars. And then there were 6 young men, well, youngish. And so we set off, and we decided that we were going to go to every country in Europe, driving around for about 6 weeks. And now I think PhD students who start their degree, if they were to, after 9 months, go off for 6 weeks holiday, I think their supervisor might frown upon this a bit. Anyway, we set off, and the goal was, we thought the college, Corpus, offered grants for people who had special ideas for going on an expedition. And in order to help pay for the trip around all the countries in Europe, we wrote a proposal to Corpus. And we said we were going to go to see the temples, the Abu Simbel temples in Egypt, because they were about to be moved, because the Aswan Dam was going to flood it. And they have been moved, so you can still go and see them. So we wrote this proposal, and we got £100 from Corpus to help us to go all around Europe. And we did that, parked our cars in Athens, and we got the boat, and we were heading towards Cairo, but the money ran out in Crete. So we spent about a week on Crete and then came back. And so the report about Abu Simbel was never written. But Corpus didn't seem to mind, So we're still planning to go, actually, and we might go next year, actually. That would be a good one. And then the last story about Corpus, there was the old college, and there was the Leckhampton, and there was, there's a lot of contact between it. But in those days, and I don't know quite what it is now, but it used to be that the May Ball would be held in Corpus one year, and then in Leckhampton the other year. It alternated. And so the year that I was here, there was a group of us. We decided we would be the ones organizing the Leckhampton summer party. And there was a lot of, we thought we better make sure that the wine was good. So we spent months going around all the, tasting the wine suitable, for the party. And then 2 of the other students there, who were called Noel Purdom and Graham Salomon, they were more arty types. They decided there should be a play a Greek play performed on the patio outside the old house. And this play was called Katsurbos. You can look it up, actually. And everybody had to be part of it. And my role, I had to be Katsurbos, who was, it was a minor role, but it was the title role. So those are my main remembrances from the lecture. And then I went off around the world and came back at Cambridge. Okay, so I'll show you a couple more slides. So that's the old house. This was the building that people live in now, and I had a room on the second floor there, and with the Henry Moore statue. And then, of course, we all went swimming in the swimming pool. It's changed since then, I think. It has a lot of leaves in it, and I think maybe the shape's been changed. I don't know, or maybe it's just the paint on it. Okay, so that was my little mini introduction about the connection to Leckhampton. Okay, so coming back now to electron microscopy, lots of people know about microscopy. They know about using an optical microscope. 300 or 400 years ago, the lenses were developed. And the only difference is that electron microscopy uses electrons instead of light to take pictures of molecules. And because the wavelength of the intrinsic wavelengths connected to the electrons is much smaller than light, you can go to much, much higher magnification and see atoms rather than cells or things like that. However, electron microscopy hasn't been used a lot for determining the structures of all the molecules in biology. And in 1950, really none of the structures that we know so well now, there was no knowledge about it. And so people had to try their best with other methods. And so the ones that, and I think you can just about see the red dot from the laser pointer, the main method people were trying to use to find out the structures of biological molecules was X-ray diffraction. And the work in Cambridge was led by Max Perutz and John Kendrew, who were the two people that I talked to when I came down to be interviewed to do a PhD in Cambridge initially. And I'll say a little bit about that first. Other people, including Linus Pauling, who had a Nobel Prize for his work, developing models for the structures in biology. They did it by putting together information from all sorts of distilled from other people's work and from the literature, from their own work, and then building a model which was just a hypothesis, and then you had to test the hypothesis. And then in between the early use of X-ray diffraction, X-ray crystallography, and model building, the chemists came in with much more powerful methods that were not destructive, of using nuclear magnetic resonance methods to indirectly find out about how the atoms were connected together, and then use a computer then to build a model. So I thought, actually, to give you a sort of feeling of the power of electron microscopy, I'll just show 2 or 3 slides about these other methods first. So one of the most famous uses of X-ray diffraction came from these pictures on the top right, which were taken by Raymond Gosling, who was a PhD student with Rosalind Franklin at King's College in London. And they took these pictures 1951, 1952. But Jim Watson and Francis Crick, putting together all sorts of other information that was distilled by talking to people, reading the literature, came up with their model. And so that was one of the earlier successes of a combination of X-ray diffraction with model building. And then a second one was the work of Linus Pauling, who was a chemist at the California Institute of Technology in Los Angeles. And he had done X-ray structures of individual components of the protein molecules, amino acids and dipeptides, 2 amino acids connected together, and used knowledge that he gained from that to build models of helical arrangements of the polypeptide, and also side-to-side arrangements, which is the beta structure. And that was just one of many models. And with Corey, they had a number of papers before anybody else. And the most famous one, though, is his alpha helix, and this is him with his model after his 1954 chemistry Nobel Prize. And then the people in Cambridge that I mentioned before, John Kendrew and Max Perutz. So I came in February 1966 to be interviewed, and those were the two people that I talked to. And they said, so Kendrew wasn't, they were not cross-examining people. They just wanted to recruit people, anybody almost, because they, wasn't all that popular then. So he said, have you got any questions? So I said, well, I come from physics and maths, and I've been wondering whether you need to know any chemistry or biology to become a structural biologist. He said, I wouldn't worry about that. You can pick up chemistry and biology as you go along. And, that is true, because every 2 years, methods and things change. And so you have to learn, even if you're already trained, you have to keep learning and so on. So these were the crystals of myoglobin from the sperm whale that John Kendrew made, and Perutz was making in parallel similar crystals of hemoglobin, and he was a bit ahead. So Kendrew was a PhD student of Max Perutz, with Lawrence Bragg being the sort of overarching umbrella above both of them. And they tried something like 25 different species before they get those beautiful crystals. And then by using X-ray diffraction, they got first this model, which they said this was the first protein structure was the ugliest molecule in existence. But these rod-shaped features that were made out of plasticine, so modeling clay, the distance between this one and the next one, it's about 10 angstroms, so 1 nanometer, and that was exactly the dimensions of the alpha helix that Pauling had predicted 2 or 3 years earlier. So it became clear that many protein molecules would be made up like this, and this was the data they got, and this was the initial structure, and then with a bit more work, another 2 years, they were able to get a very high, and then build a model with all the atoms in the myoglobin. And this made a really big impact, actually. So, and also there was no computer graphics then, and no movies and so on. So they persuaded an artist, Irving Geis, to do a watercolor, and he was very clever. He moved it around slightly so none of the atoms overlap, which of course they do in reality. And this painting was now bought and is hanging in the Howard Hughes Medical Institute in near Washington. So that was the X-ray, and then one last slide about other methodologies. Kurt Wittrich, who was a chemist from Switzerland, using the method that another guy, Richard Ernst, had developed, Fourier transform NMR, got spectra like this, very complicated, but apparently you can interpret all those little spots, and you end up with what's on the bottom left, this structure, but it's completely indirect. So there's no structure in this, just information, and you end up with a structure. So the great thing about electron microscopy is much simpler than all these. You just take a picture, and there you look at it, and that is the molecule. So I'm going to just give you a couple. So the electron microscope itself didn't exist till about the 1930s, and a man called Ernst Ruska, so have I got my pointer here? Yeah. In 1933, put magnetic lenses with an electron beam and made the first images. And his brother, Helmut Ruska, took these very first pictures of biological structures. So this is a bacterium, and these are little bacteriophages with tails on them that bind to the bacteria, inject their DNA or RNA, and then infect the bacteria. And then a little bit later, another method, early method of doing electron microscopy with biological structures, instead of staining them with heavy metals, these are shadowed with heavy metals. And this is Robley Williams a bit later on. And again, they liked these bacteriophages because they were at that resolution that they had then, you could recognize something. And then some time later on, at MRC Molecular Biology, the people who had a crystallographic background, and this was Aaron Klug's group, and David DeRosier was an American postdoc working with Klug in 1968, took pictures like this of the same bacteriophages that were the earliest images taken with the electron microscope of biological structures, but better now, higher resolution, more nicely preserved. And then they took the image of that central region, the bacteriophage tail, and then they said, okay, that is one view of the structure of that biological part of the virus. However, because it's a helix, you can rotate it by a certain amount, and then you get the same view again. So from this one view, it turns out you can work out the three-dimensional arrangement of the protein molecules, although it's not the protein molecules you're looking at, it's the stain that's surrounding it. And they got this three-dimensional model of the bacteriophage tail. And then a little bit later, Tony Crowther, who was trained in maths in the maths lab here in Cambridge, applied similar methods to these icosahedral viruses. And this is tomato bushy stunt virus, done about 1970. So that was the beginnings of three-dimensional analysis of two-dimensional images in the electron microscope used for biology. So I got involved with electron microscopy, not really as a method of first choice. I was really trained as an X-ray crystallographer. So I worked on, when I was here doing my PhD, I was a crystallographer joining a group that was working on the third protein structure. So it was myoglobin, and then lysozyme, and then chymotrypsin was I joined the group that was working on that. David Blow was my supervisor, and I didn't discover this till later, but David Blow himself was an undergraduate here and a PhD student here in Corpus. So, yeah, but not in Leckhampton, because that was in the 1950s. So the way I got involved in electron microscopy was, sorry, I have to go back. I met Nigel Unwin, who was a New Zealander who came to Cambridge to do a PhD in metallurgy and material science. And then he was recruited as a young group leader from material science, where he did his PhD, to MRC lab in Cambridge. And he was an expert in electron microscopy. And I'd been working on this bacterium, Halobacterium salinarium, which has a membrane around it. And if you freeze the sample and crack it open, this is called freeze fracture electron microscopy, you see a pattern of all the protein molecules in the membrane. And there are these little patches that are different from the rest of it. So most of the membrane has hundreds of different molecules in it that are responsible for the bacteria signaling to the outside world, transporting things in and out. But this little patch here is just one protein that has a purple color, absorbs light, and then that light energy is used to power the bacteria so they can swim around, it can metabolize amino acids and so on. And if you isolate those patches and put an electron beam through them, you get a diffraction pattern just like this here, just like the X-ray patterns. And so for many years, we did what's called electron crystallography, which was similar to X-ray crystallography. We weren't doing electron microscopy. And then at that time, which was 1975, we managed to get this we had something like 30 of these diffraction spots. The ones at very low resolution were measured and converted, and we got this model. And in other words, in that patch of membranes, there's about 100 molecules this way, about 100 molecules that way, so about 10,000 molecules in that patch. This is one molecule cut out of the calculation using the data from that image. And these features, these rod shapes, the membrane would be horizontal in that view. There are 7 of these rod-shaped features, and those are alpha helical stretches of the polypeptide, the same ones that were in myoglobin and the same ones that were predicted by Linus Pauling. And so we both went on to work on other membrane proteins and at higher resolution. And after about another 15 years it took, actually, because there were a lot of interesting problems to be solved that didn't make a lot of impact, actually. And of course, we were reviewed by the funding agencies. And I remember one particularly critical referees report who said, and it was before that they said, none of these methods are going to work. So, this should not be funded. But fortunately, there were other people doing good work, and the lab was being funded. And so we were able to go on without publishing any papers for many years, actually. But eventually we got this, this higher resolution map, and these are the same 7 helices. So 1, 2, 3, 4, 5, 6, 7. And then the chromophore, the purple color, it's vitamin A aldehyde, binds to the protein. And now you see not just smooth rod-shaped features, but they've got side chains sticking out. This is phenylalanine, tyrosine, tryptophan, and so on. So we were able to build an atomic model in 1990, And that showed that you could use electron microscopy or electron diffraction to get biological structures. But the real power didn't come till a little bit later, and it took a long time. But in the year 1999 or the year 2000, Tim Baker and I wrote this review, which was in the crystallographic Bible called International Tables for Crystallography, and there were about 80 or 90 chapters, all about X-ray diffraction, X-ray imaging, a little bit about neutrons, and two out of 90 chapters were about electrons. One on electron diffraction, which is what we've been doing, and a second one on electron microscopy. And Tim and I we wrote this chapter, and we thought, well, nobody knows about this. So we made this slide, and we thought, well, if we make it a good slide, we can show it to people forever, because it's an introduction. So what is electron cryo microscopy? So these are 4 images recorded in the electron microscope of different biological structures, different types of biological structure. And in the year 2000, 1999, 2000, this was the best you could do using images like this. And the first one is an electron micrograph of a thin film of amorphous ice, rapidly frozen, and I'll show you a bit about that in a moment, done by the group of Joachim Frank, who was one of the people sharing this 2017 Chemistry Nobel Prize. And using images like that, they were able to get an 11 angstrom resolution structure. So not enough to see the spacing between alpha helices, but it was enough to see stretches of RNA and single and RNA double helix, and also to see where the large subunit of the ribosome and the small subunit are. And so that was a model of the ribosome from 20 years ago with technology the best you could do in the year 2000. And the second panel is hepatitis B virus cores, and this was done by Bettina Botcher, who was a postdoc from Germany originally at MRC Lab, working with Tony Crowther, who'd worked on these other icosicroviruses. And we had invested in some technology, not as good as we have now, and she was the first one to get images of single particles, in other words, molecules or molecular structures that were not arranged in a crystal, and digitize them and process them and get a model where the resolution went below 10 angstroms, below 1 nanometer. So this is a 7 angstrom resolution picture of hepatitis B virus cores, and it has these protrusions, and you can see little ridges along them, and each of those protrusions has 4 of these Pauling alpha helices in a little bundle sticking out from the surface. And you know, that was a lot of work. She worked for about 3 years to get this structure. Ron Milligan was a student, PhD student, with Nigel Unwin in California, and he worked on the fibers from muscle. So this is fibers of actin, which is the core of the thin filament in muscle. And also in this image is tropomyosin that helps to hold the actin together, and the subunits of myosin that have been purified biochemistry and then added to the actin, like muscle in rigor, where it's gone solid. So you've got the heads of myosin that normally form the thick filaments that attach to the thin filament and pull the muscle along, and you get your muscle contract. So even with a very low resolution, they were able to find out interesting things about how muscle works. And then the last picture is another structure based on electron crystallography. So our work on Bacteriodopsin, that was the first one. The second one was this light harvesting complex from green plants, which Werner Kuhlbrandt, who'd been a PhD student with Nigel Unwin in the lab and went off to Switzerland and then to EMBL, and they got this model. And these are the molecules. Each of them has about 13 chlorophyll A or chlorophyll B bound to them. And if you look out and you see the grass and the trees, the green color is this molecule there. So this was just a spectrum of other people's work, single particles with no symmetry, single particles with symmetry, helical arrangements, one-dimensional order, and then two-dimensional order, showing the range of specimens. And it's what you use the same electron microscope, the same method of making the specimens, to extract the information about the structures. Okay, so alongside this work, Jacques Dubochet, who was a PhD student in 1970 in Switzerland, working on biological structures using stain, he was recruited from Switzerland to be one of the first young group leaders at the European Molecular Biology Lab in Heidelberg. And the person who recruited him was John Kendrew, who'd worked on myoglobin here. And Kendrew became the director and recruited a lot of young scientists. And Kendrew had a lot of vision, and he said, well, there's a lot of power potential in electron microscopy, particularly if you can develop methods of freezing specimens so that they're kept in the microscope cold and you don't have to use these heavy metal stains. And so Jacques worked for about 3 or 4 years, from 78 to about 82, developing methods for preparing samples that had been frozen so that you could then take images of them. And this is his apparatus. So this is a pair of forceps, and on the end is a 3 millimeter diameter electron microscope grid. And the method of preparation that he developed, and which everybody uses now, so this is now 40 years later, and it's the same method. You put a little drop of your sample on this thin grid, blot it with filter paper, and then release this catch and plunge it down into this little chamber here, which would have liquid ethane just above the temperature of liquid nitrogen. So minus 196, something like that, minus 185. And then the ice, the thin film of ice, with your viruses or ribosomes or protein molecules in it, gets very rapidly frozen, and you don't get ice crystals, and you get really nice pictures. And so this one is a gravity-fed plunge freezing device. And then they thought, well, maybe we should make it plunge faster. And so they have this now elastic band-powered freezer. And they work, and they're very reliable. But of course now, if you want to do plunge freezing, you have to spend £100,000 and you buy a computer-controlled, humidity-controlled, temperature-controlled system. And I have to say, these ones are more reliable than the modern ones. But anyway, that was Jacques' early 1980s contribution. And this is a picture from a review he wrote in 1988. But one of the earliest pictures he took is this picture, which is adenovirus, which is still around. And, you know, adenovirus is now one of the vectors for some of these coronavirus vaccinations. And there's an interesting story about Jacques here. So he was a PhD student in Switzerland, recruited as a group leader by John Kendrew, and they have a 9-year limit at the EMBL. So at the end of the 9 years, Kendrew retired and the new director came. And the new director's first act was to sack half a dozen group leaders, the ones he thought weren't contributing very well. And one of the ones he sacked was Jacques Dubochet. And that was before he took this picture. And the great insight that Dubochet had was to decide to take an image of the virus that Leonard Philipson had worked on for his whole life. So once Philipson saw this, Dubochet was reinstated and then stayed at EMBL until about 1988 when he wrote that review. So that was 1984, and this picture was published on the front cover of Nature then and made a lot of impact because people realized if you can take such beautiful pictures of a virus, no stain, no metal, no shadowing, and this is the amorphous ice made by the Dubochet plunge freeze method, and most of them are beautiful. Some of them have been damaged by the blotting and the freezing, and these little dots here are some of the subunits that have fallen off. But generally, it's pretty good. But the methods for dealing with information like this were not available in 1986 and so on. So it took them from 1984 until 1991. In this picture here, so this is an image, three-dimensional image, using the similar methods that I showed you earlier, made by Phoebe Stewart, who is an American postdoc at EMBL, took images like this, and it took 7 years to go from this to this structure. And it's very, very low. You can't see any helices. It's very blobby and so on. This is 1991. And so that's how it stood for many years. And in our field, the proper structural biologists namely the X-ray crystallographers and the NMR people, they said, oh yes, these electron microscopists, they are blobologists because they only get blobs. And actually that was true for about 10 or 20 years. But it took, so from 1991, which were on the left, I've lost my laser pointer. Anyway, the left-hand one, 1991, the right-hand one is 20 years later. And this picture is the same adenovirus taken with technology that had been developed. So better microscopes, better vacuums, brighter sources, everything's more coherent. And so in the virus now, instead of one blob that you can see here, you can see much more finer structure in the viruses. And when those were processed in the computer, these images produced a structure like this. And instead of it being at a resolution of 35 angstroms, it's now 3.5 or 3. So 10 times better resolution. And that's in 1 dimension. In 3 dimensions, that's 1,000 times more data. And so it's now, it's less blurry, it's higher resolution. And then when you look inside the protein, you can see the stretches of polypeptide here. And this doesn't have any alpha helix in it, but it has beta structure, which was another of the structures that Pauling had predicted in 1950. And occasionally, resolution is not quite high enough. You can see the beginning of the amino acid side chains that allow you to interpret the density maps in terms of the sequence of the protein that you're interested in. And that's how it stood for a while until about 10 years ago, new detectors came in. And, our group and other groups all around the world had been working for, we started about 1990, but by 2012, some new electronic detectors came in using electronic camera technology, just like you have in your phone. So now if you buy an iPhone 14 Pro, it will have 3 CMOS detectors in it, and they have between 12 and 50 megapixels of data. So in the year 2012 or so, we started to get new detectors with technology in the electron microscopy. It's about 10 years behind the technology in phones and digital cameras, because they have a big market, whereas the electron microscopy market is very small. So, each sensor in your phone, they can afford to spend 10 or 20 million developing it, because every year they're selling, hundreds of billions of dollars worth of phones. Whereas in the electron microscopy area, they're selling 5 or 10 micro, they just cannot afford it. But if you know anybody who's, got an excess of 10 or 20 million, making better detectors would be a good thing. Anyway, this was film that we've been using since 1960 to 2010, and it detects about 1/3 of the electrons. So the detective quantum efficiency is the proportion you detect, about 1/3, and then at high resolution it gets very, very bad because the localization is not very good. All these new detectors were better, and now, of course, 6 or 7 years later, more money has been put in. We're now up to about 100%, going down to about 60% here. So they're much better now. So these new detectors came in, and they had one advantage was they have a higher detective quantum efficiency, but a second advantage is, just like in your phone, now you take a photo, and actually, if you've set it up right, the photo has 4 or 5 seconds of movie before the photo and 4 or 5 after it. So you can, if somebody blinks, you can just go, 10th of a second further on, and you've got it. So these detectors for electron microscope are the same. So you can process the images, and if there's any blurring of the image due to radiation damage or charging, you can remove that in the computer afterwards. And also, the radiation damage can be programmed to get the best part of the image at the beginning. So there are many advantages, and these new detectors, it only looks like it's a factor of 2 or 3 here, but actually the gain was more like a factor of 30 or 40. So this is a very big jump forward in the technology, and that allowed much better images and much better structures, and that's what produced what they now call the resolution revolution. So you could get structures before, but they were blurry and they were blobby. But now you can get structures way out beyond the atomic resolution and so on. So I'm just going to show you 2 or 3 of these to give you a flavor of it, and then we'll come to a kind of overview afterwards. So this is one of the structures that made a lot of impact when it was being done. So let me see if I get. Alexei Amunts was a postdoctoral scientist who came from Israel to work with Venki Ramakrishnan on ribosomes. And Venki had been doing X-ray crystallography for about 20 years and had determined ribosome structures and has a Nobel Prize in 2009 for that work. But he was always interested in using the electron microscope to do more difficult structures. And so this image here is a picture of mitochondrial ribosomes. And so the way you get mitochondrial ribosomes is you start with a cow, and then you get the heart out of it, you grind it up, and then you get the mitochondria out of the heart, and then you break it open, and then you look for the mitochondria that are inside, the ribosomes that are inside the mitochondria. But there aren't many of them, and it turns out it's very difficult to purify them because the rest of the cells, the rest of the heart, the cytoplasm, they have many more ribosomes. And so although Alexei worked for about 2 years to try to purify mitochondrial ribosomes from beef heart, he never got it pure. He never got crystals. And so another postdoc, Xiaochen Bai, came along and worked with Shor Sherez, who wrote some computer programs. So there are 2 groups at the LMB, a ribosome group and a computer image processing in electron microscopy group. And then 2 talented young postdocs took images like this and picked out all the particles and put them into a computer program written by Shores called Rely On, and got initially a structure like this one with all the particles. But in Rely On, you can say, I think my specimen is a mixture of different molecules. I haven't purified them properly. And so you can ask for 6 or 10 or... So he asked for 6 classes, and it turns out 4 of them do have mitochondrial ribosomes in them. 1 of them has cytoplasmic ribosomes, and 1 of them they weren't sure what it was. But you can see in these classes, they're not all intact ribosomes. These 2 are intact, slightly different conformations, but these ones have lost the small subunit. But one of the other features that you can do, because you have an image of it and you're looking at the structure, it's a bit noisy. You can focus on the large subunit and find the orientation of all the particles based on that one part of the structure, and then get a really accurate, high-resolution image of the part you focused on. And then from the same data, all with just a little bit more computing, you can focus on the small subunit and then get an accurate picture of that. And by doing that, they got the structure of the entire bovine mitochondrial ribosome here. And I'll just show you one bit of the map. This is a little bit of RNA, Watson-Crick RNA double helix, just like the DNA double helix, but it's RNA with the extra hydroxyl. And these are the base pairs, 3 of them. And then this is a top view with the hydrogen bonds that do recognition between the bases. And you see that you collect the data, you process them, you get the structure, and then there are hundreds of these bases in the ribosome structure. And then on the right are parts of the protein components of the ribosome. So here is an alpha helix and 2 loops with a magnesium ion and so on. So with one experiment, you get the structures of all the different parts of the ribosome, all the different parts of the protein, nucleic acid, and so on. And even now, this is the only way of getting that structure using electron microscopy. You certainly can't do it by the other methods that there are around. So then a second structure, which I think is very good, and obviously there's a sort of corpus angle to this. These are electron microscope images of mitochondrial complex 1 that Judy Hurst has worked on for many years. And it was Judy's initiative. She had observed what people were doing by electron microscopy, and she came over from the MRC mitochondrial unit in Cambridge to the MRC molecular biology unit one day about 2013, 2014, and said, I think what we've been doing, trying to make crystals and so on, is a specimen that would be suitable for electron microscopy. And she worked together with her postdoc, Jaipen Ju, and a postdoc in our lab called Vinod Kumar. And Jaipen's back in China, Vinod's back in India. But within a very short period of time, they had this three-dimensional structure of these mitochondrial complexes, one of the biggest structures in the membrane that takes redox energy from NADH and pumps protons across the membrane, creates a potential which can then be used to make ATP. And so this structure, contoured at a high level, shows 8 of these brown features. These are iron-sulfur complexes. And the way it works, the electron is fed in at the top from NADH. It goes down this chain and reduces a quinone in the membrane. And then something happens in the membrane to pump protons across it by mechanisms that are still being worked on by Judy's group and by, we had a seminar earlier in the other Marquee this morning from one of the people in Judy's group. But with a little bit more work, Jaipen and Vinod, at a lower contour level, that's the lipid part. The protein is in the mitochondrial lipid. With a little bit more work, in another year or so, in 2016, they were able to make a model of all 45 different polypeptide subunits that make up that complex. The blue color are the core subunits, for which there was a homologous structure done in a thermophilic. So that's a much more stable protein molecule from bacteria that another group in the then mitochondrial biology unit, led by Leo Sasanov, had done by X-ray crystallography. And Leo is now leading a group in Vienna, and now Leo as well have switched back to using, and everybody is now using electron cryo microscopy to determine these structures. But all the red subunits were determined by the cryo microscopy, and there are 31 of these supernumerary subunits that are in higher organism, sort of mammalian mitochondrial ribosomes, whereas there are only 16 of these core subunits. A lot of them are bigger from lower forms of life, but also in higher forms. So these are 2 really good examples, but I'll just show you 1 or 2 more things. So another question came up was in these images. So on the top left is an image that Miriam Koshai, who was a PhD student in Martinsried, just outside Munich, led by her supervisor Radostin Danev, with Wolfgang Baumeister as the director there. And Miriam, on one day, 1 week, Thursday or Friday, she said, I think we should try to take images of hemoglobin, the molecule Max Perutz had worked on by X-ray crystallography. And we always thought, actually, it would be wonderful if we could show Max the structure of hemoglobin determined by another method than X-ray crystallography. And so what happened to Miriam, against the advice of her supervisor, just decided to do it herself and took this data over 1 weekend, made these 2-dimensional classes, and ended up with a 3D model of all of human hemoglobin with the heme group and the proximal and distal histidine. This is where the oxygen binds in the oxygen carrying hemoglobin. So it turns out you can do structures that you couldn't do by other methods, but you can also do a wide range of structures that you can do by other methods. And this is still quite a triumph. So then if you take a step back and ask, well, since it works so well on these 2 or 3 things that I've shown you, what about the rest of the world who've now piled into using this methodology? So here is a plot of the number of structures in biology determined at a sufficient resolution so that you can build an atomic model by the different methods. And so the methods that are here are X-ray crystallography, electron microscopy, and nuclear magnetic resonance spectroscopy. And you can see in the year 1999-2000, there's nothing from electron microscopy at all. But there's thousands every year being deposited done by X-ray crystallography, and NMR was beginning to compete, so maybe 20% as many. And that's because if you couldn't crystallize your protein, you couldn't determine the structure by X-ray crystallography, so you had to use other non-crystallographic methods. That was NMR, which grew and grew and grew in popularity. It peaked in the year 2007 with about 15% the number of atomic coordinate depositions per annum as in X-ray crystallography. So that's this peak down here. And you can see there's still nothing until about 2012 from X-ray crystallography, from electron microscopy. So one plot that we like to do is to compare now what is the impact of all the different methodologies on an annual basis. And it's quite illuminating in terms of where did the advantage come from. And so you can see from, let's say, the year 2000, there were 1,000 or 2,000 times more structures being determined using X-ray crystallography because it was so powerful, and they have all these synchrotrons, and it's been really streamlined. And then better microscopes came in, and that dropped from about 2,000 or 3,000 to 200 or 300 times as many. And it sort of stayed like that until 2012, which is when these new detectors came in, the ones that are based on digital camera technology. And now, on a log plot, so exponential growth, it's completely linear, actually. And so the predicted point where in 1 year there'll be the same number of structures done by the two methodologies is, it turns out, it's August 2024. Of course, we worry about these exponential growths. It's very difficult to keep them going. So one comparison that's quite good is to look at the absolute number, no comparison now, structures determined by X-ray crystallography. You can see there were not many per annum, but then the new synchrotrons and better ways of freezing crystals came in, and there was exponential growth for about 10 years with a factor of 100 improvement in productivity, and then it sort of plateaued. And then NMR grew and grew and grew, and then peaked, and then, NMR is used for other things now rather than de novo structural determinations. But the cryo-EM didn't kick off till about 2014 or 2013, and it is still in exponential growth. And we have looked at certain areas of structural biology, molecules in biology, and many of them are done very well by cryo-EM. So, for example, in our lab now, the crossover happened several years ago. So we're pretty sure now that this exponential growth will go on for at least 8 or 10 more years, because certain sub-areas have continued to do that. And so the rest of the world, at some point, but our current worry is we'll run out of some other factor will come in that limits it. So just like in other areas of science, so for example, the bottom here is Moore's law for computer technology. How many transistors per square millimeter? And they've managed to keep going for 50 years, doubling it every 2 years. And that's an increase of 10 or 20 million fold. So, the computers we used to use in the 60s, you can now buy, tens of millions or billions of times more power for less money than you had to pay in the 1960s. But in order to do that, the computer field, of course, has a much more. It's useful for everything, and it permeates all of life. So they've got a lot more money than the electron microscopy community has. But if you can identify a likely bottleneck, let's say 5 years down the line, then you can start developing the technology that's needed to make finer and finer features in the computer chips. And that's what they've done. And so one of the things that we worry about now is what are likely to become the rate-limiting steps that make electron cryomicroscopy as a method in structural biology plateau in the future. And then can we overcome them using similar approaches to the ones that the computer industry have done? Okay, so that's more or less giving you an overview of what is cryo-EM and what, why it's so useful. But I thought I'd show you a few examples now of how its impact has spread into other areas. So one of the many structures that we looked at, which produces pretty pictures, was this image, this top left. This is a picture of human red blood cell catalase, which removes reactive oxygen species like H2O2 and so on, and stops your body being damaged. So that's when we first took a picture. It forms these ribbons. And then we thought, well, we need to get rid of these ribbons because we need to get separate molecules in all the orientations to get the 3D structure. So we added some detergents that stops the molecule interacting with the air-water surfaces that get produced when you blot these thin films using the Dubochet method. And we now get two views. The top view of a thin view of the molecules because it's been squeezed out by being blotted. And then the same views as here, but not in ribbons. And then eventually, with a different, better detergent, we now get all these different views. And by doing that, you get the structure of the human erythrocyte catalase with the cofactors, the non-polypeptide molecules that are in the structure here. So that this is the heme group, just like the heme group in hemoglobin. This is a cofactor that's bound on the surface, NADPH, with 3 phosphate groups. That's these yellow with the red. So you get an atomic model of the protein and all the cofactors in it. And then these are just helices and sheets down below. And then a second example is a structure that Vinod Kumar worked on after he went back to India, and it's called PAZ. It's a phenylacetic acid degrading enzyme, so it's a detoxifying molecule. And these are 3 structures done by electron microscopy. It has a 3-fold axis, and the structures have different ligands bound to them, just like the ligands I showed you. So one ligand, the other ligand, and then both ligands. And that's the overall structure. And then you just magnify the ones that are in red, and you can see that the density features can be interpreted in terms of the structures really quite nicely. So that then led into the interest of the pharmaceutical companies. This slide comes from work done by the company Astex on the Science Park, and they've been around for 20 years. I think Tom Blundell was involved with starting it. Haran Jyoti is the CEO. And they've been doing X-ray crystallography, determining structures of proteins, designing better drugs, and then these drugs make their way through into therapeutic medicine, phase 1, phase 2, phase 3, and then they're curing people of different diseases or physiological problems. And so they developed an interest about 4 or 5 years ago. And they said, I wonder if cryo-EM can be used to help design better drugs. And so they asked first, could we suggest something they could practice on? So we suggested this beta-galactosidase molecule, which is about half a megadalton, so half the size of that mitochondrial complex 1. And they did about 60 data sets in 60 days, 1 data set per day, using a microscope that they bought from the companies, and it was installed in the nanoscience department of Cambridge University, shared with 5 pharmaceutical companies. Astex was the lead company, but there were other ones, GSK, AstraZeneca, all the ones that you've heard of, and they're all now doing it. They used to be doing, they did drug design by guesswork and chemistry until about 1990. Then they used X-ray crystallography, and all the big drug companies and lots of the smaller ones, maybe 30 of them, are using structure knowledge about the drug target to design better drugs. And so now about half of them, probably 15 or 20, are using electron microscopy now just as a tool for doing dozens or hundreds of structures with different ligands bound. And these are 3 structures done of beta-galactosidase with different inhibitors bound to it. And then the bottom one is a real anti-cancer drug target with 2 drugs bound to it. But these are all ones that they feel are not their top candidates. They're the ones that they were willing to publish. And so this was published about 2 or 3 years ago, probably, let's say, coming up 2.5 years ago in Drug Discovery Today, showing that you can use electron microscopy for drug design. And then 2 other ones. One of them is So there is another family called G protein coupled receptors, which are responsible for an enormous amount of signaling, lots of disease processes. And Chris Tate and I in the MRC lab got involved in doing this work many years ago, actually. And one of the startup companies from the MRC, Heptares, does a lot of this work. But in Chris's lab, with a guy, one postdoc, Javier Garcia Nafria, came from Spain and took images like this one on the bottom of the adenosine A2A. It's one of the family of receptors. For adenosine, you can inhibit it with caffeine and so on. And it does signaling, and this particular structure, an inhibitor of that, somehow or other helps prevent certain types of cancer. So that's in drug development. But here's the image. Here are the two-dimensional classes, and here is the map and the model that they built. And if you look carefully in that little square at the top, you can see the ligand, which is N-ethyl carboxyadenosine, which is the ligand that binds to this and can activate it. And then a second one is another G protein coupled receptor called 5-hydroxytryptamine 1B, serotonin also, and this is one for which this drug donitriptan and other homologs are currently used for migraine treatment. So again, they're trying to make better drugs. So that's two examples of, let's say, medically important things that have been, let's say, cryo-EMs beginning to have some impact on it. And I'm going to show you two other things now. So one is, once you've bought an electron cryo microscope, which costs a lot of money actually, a few £1,000,000 each, and so one of the things is to try to make them cheaper so that every student can have their own. That's the goal. But once you've got it, you can look at other things than highly purified molecules with drugs bound to them in a thin film of amorphous ice made by the Dubourg method. And this is an example of a different type of use of the cryo-electron microscopy. They call cryotomography, and you don't do any purification. You basically take a thin slab of a cell, either made with a diamond knife, cutting it up after it's frozen, or they have what's called focused ion beam milling. And this is from 20 years ago. So this is Ohad Medalia, who is a postdoc with Baumeister, the same person, the German Max Planck leader in Martinsried. And this was published in 2002. And this is an intact cell. The blue are membranes of the cell. The red are filaments of actin. And these sort of greeny yellow things, they are the ribosomes. And in the year 2000, that's the best you could do. But if you now jump forward by, let's say, 15 years, you now get, with the new detectors and the new microscopes, you now get much better quality micrographs here. And so this is one taken by Julia Mahamed, who's a postdoc in that same lab. She's now a group leader at the European Molecular Biology Lab, where Kendra and Dubochet worked. And when you look through these maps and render them by segmentation, you see the same features you saw before, but now in much more detail. So here are the membranes. These are the nuclear pores, which have 8, let's say, domains, 8-fold symmetry. These are the brown is the actin filaments, the green are microtubules, and then the ribosomes now, you can see, they have two colors now. Blue is the large subunit, and yellow is the small subunit. So you can find in the tomogram now individual ribosomes, find their orientation. Are they bound to the membrane? Are they bound to in a stretch of adjacent molecules? So much higher resolution. And the hope is that all of the methods will be able to be improved in the future. So this is, although a lot of good work has been done, there's still a lot of unfulfilled, untapped potential coming up. So I sort of finished with one last thing is, of course, we've all been, or many of us have been locked out of our labs for 2 or 3 weeks, and everybody was told they could only go to work if they worked on coronavirus. And so in that database I showed you of all the protein data bank, there was actually a spike in the year 2021. And that's because all these coronavirus structures, drug targets and tomograms and so on. And so this is, on the left, are actually, that's cheating. These are both artists' impressions. This is Wikipedia's explanation, beautiful, clear, with the spike glycoprotein sticking out from the outside of the virus, and then the RNA and the RNA binding protein in the middle. So this is the S protein, and this is the N protein, and these are, so the S protein is used in the vaccine, and the N protein is used in these lateral flow tests where you find out if you've been infected, because you can only have the N protein if you've been properly infected. And then here's an artist's rendering of a rather beautiful, I don't think there's any virus quite it. This is the best quality coronavirus that you can imagine. But on the right is the real thing, and this is from work John Briggs did when he was a group leader here. Now he's gone to Martinsried to be a German Max Planck director. And this is a real electron cryotomogram of, obviously they're formaldehyde inactivated viruses so that you don't get infected. But it's got these spikes, and they change their structure when they bind, and then they fuse, and then they inject the RNA into it. And so there's been, a lot of groups have been doing tomography, but some groups have also been doing the single particle work. And so here is a picture of the spike trimer, and that is the bound angiotensin converting enzyme, which is the receptor on your nasal passages and in your lungs and so on, for the adenovirus. So the adenovirus binds to this and then fuses and injects the RNA and so on. And so there are actually 5,000 structures of similar, this with different antibodies and similar things that have been done by the community, all in the space of about a year or two. So, the cryo-EM is making an impact across the board, actually. And so now it is, I have to say, it is the method of choice for anything to do with structure and structural biology. So I'll finish just by saying these are the things I've picked a little bit of our work, but mostly work from other people, starting with Dubochet's adenovirus and Hong Chu's improvement on it, the work from Alexei Emmons and Xiaochen Bai, Vinod Kumar, led by Judy Hurst, and then hemoglobin, catalase, and so on. So lots of structures. And then the final one is people that I've worked with over the years, and I wouldn't be doing this if I hadn't met Nigel Unwin. He gave a talk in 1973, and it was clear he was thinking, even though it was with Stain and it was the early days, he was thinking in his mind about all the problems that needed to be solved to make the electron microscopy become really powerful, actually. And so if I hadn't met, I would still be doing something different. And then these are all the people. Joyce Baldwin is retired now. She's in Cambridge. Tom Seska went to work for a pharmaceutical company and just retired. David Agard is now the leader of a big structural institute in UC San Francisco. Bob Glaser at Berkeley. Jean Lepol in Paris. Fritz Emlin retired. He was in the Fritz Haber Institute in Berlin. We worked a lot with him. He and Derek, both in Berlin. Ken Downing was working with Bob Glaser. These were all the people in the early days when we were say moving from X-ray crystallography to electron crystallography. And then the next row, Wasi Faruqi and Greg, and Renato and Nicola, two Italian chip designers for making CMOS cameras that go into the electron microscope. And Xiaoxia runs our this is the electron microscopy group at LMB and collaborators. And then these were people who were here and are now off in the world leading their own groups in different parts of the world. And then I didn't talk at all about it, but the recent person that we've been working with to try and make it cheaper is another young group leader at MRC called Chris Russo. So hopefully that's given you a kind of overview of how we got there. I haven't said anything about how Nobel Prize committees go about their business, which is incredibly mysterious. But nevertheless, they've made the right decision.
That was wonderful. I think we have time for just a couple of questions, but thank you
so much for such an incredible, rich talk. So are there any questions from the, and Eliza will bring the, let's say the microscope, the microphone. Yeah.
Hello, hi, thank you. Do you think that the rise of machine learning and structural prediction could be one of these limiting steps that will make cryo-EM plateau?
Yeah, so there is a difference. It's kind of like the Republicans versus the Democrats in the American election. There's two, let's say, fields of view in this. So we do have this AlphaFold 1, AlphaFold 2, and we're about to get AlphaFold 3. And so some people think it's all over. So in fact, some PhD students in their first year, they would say, should I just give up my PhD? Because you can just download predicted structures. But they're not real. They're predicted by looking for patterns from all the existing structures and with a little bit of added things in it. And so I'm in the other camp, actually. I think if you really seriously want to know something about your structure, you need to know if it's true or not. So even though you might download something from AlphaFold and say, I think this might be close to what we're interested in, you still need to check it. But the one mistake that they made was, well, no, they didn't make it. AlphaFold did their job. They did great work. They published millions of structures. They're in a data bank. You can download them. And of course, they did do what most scientists do. They do a little bit of hyping it up. They probably did a little bit more. But the one thing I thought was a mistake was that Science magazine decided to make computer prediction the method of the year. And that was okay. It's useful for everybody. You haven't got a structure, you download something similar to it, and then you can make progress. But they said they've solved the protein folding problem. And most of us understand that you make the RNA, the DNA, you make the protein, it folds up by itself usually, although there are sometimes chaperonins that help it. But the protein folding problem is to understand how the physical forces between the amino acids and water, how does that result in a structure that's energetically favorable? And the AlphaFold isn't doing that. They're just downloading several 100,000 known structures, looking for patterns, looking for correlations between distant residues to find out if they're close, and then doing a little bit at the end to sort of smooth it out. So I think it is useful, but it's definitely not going to be people. So in fact, the field of experimental determining of structures, even though there might be a predicted one for some of them, it is growing exponentially, and it's going to continue to do that. But you should each year you should look forward to say how well has AlphaFold 3 done this year compared to the competition, because a lot of people do this. So yeah, it's useful. Computers are useful in many ways, and these neural nets are particularly good at doing face recognition. And they are obviously quite good at looking for patterns and other things as well. But I think you need to be a bit suspicious and look for other evidence to confirm whether the recognition of that face, is that really the person that it's recognized or not.
Richard, first of all, wonderful lecture. Thank you so much. But with your usual sense of modesty, you haven't quite told us what specifically you did to get your richly deserved Nobel Prize. I've read the citations, but perhaps it might be a good idea. 2 minutes, just in a general sort of way.
Well, at the risk of reinforcing your statement about modesty, I'd have to say, I thought that a Nobel Prize should be given for electron microscopy development, but I spent quite a lot of years trying to persuade them they should give it only to Dubochet. So, however, they have a committee. People make nominations, and usually now they've broadened it. It used to be one person they'd give it to, and then it was 2, and then 3, and then they've limited it 3. But my feeling is that this field, I will tell you, we did do something, but it's the field as a whole that's made progress.
Yeah.
So, we've had, well, at each stage, people tried to do what we can do now, and it didn't work. So you would get images, you could see the molecules, but you'd get 20 angstrom resolution, 10 out of 10. So you have to say, why was that? And in each case, you need to do experiments to diagnose the problem. And then let's say you think, well, the electron source isn't bright enough. So, somebody who's an expert in electron sources needs to make a better one. So we got these field emission gun sources, and field emission itself wasn't developed until about 1960. So 1995, they came in. That was one of the big improvements in the hardware. And then the microscope vacuums were really bad. So I remember when Nigel Unwin tried to do cryo-EM in 1975, as you cooled the specimen from room temperature, you reached minus 100. It was covered in ice because the vacuum's bad. It's got water vapor in it at a very low level. So you could look for about a minute, and then you couldn't see through your specimen. So the vacuum had to be. So lots of things had to be improved. And then these computer programs, without modern computing power and all the memory and all the processors, GPUs, you couldn't do all this processing. And different people have done that. So this was Shor Sherez was in our lab, but there were other people like Nikhil Grigoriev who also worked in our lab, and Stephen Lucas. So it's basically, the success of cryo-EM comes from the contributions from lots of people in the field. And if you'd been, but this is true in almost every, there's the lithium batteries, for example, it's hundreds of people, but they gave a prize to 3. So they have to pick somebody who are there, they then become the flag waver for the field. So what did we do? So, well, my personal background was I was focused on finding out the structures of membrane proteins. And so the idea was to make crystals so that you could do X-ray crystallography. And then we found it was very difficult to make crystals, or if you got them, they were very bad. So then I made the transition from X-ray crystallography to electron crystallography. Crystals only 1 molecule thick. We thought that would be easier. And there are about 12 structures done by electron crystallography, 12. Whereas now there's 5,000 a year done by the single particle method, 12 over 10 years. So after about, 1975 to 1995, that was 20 years, I did write a review looking into radiation damage and what the potential was, totally theoretically, so based on first principles physics. And then the bottom line came out that you could determine the structure of anything by single particles without any crystals, provided you got good enough data. And so in 1995, we published that review, and that's had a lot of impact, and people paid a lot of attention to it. But because I had written that review, we decided we'd stop all crystallography, and we started doing the single particle work then. And then there were other problems, and one of the main ones was the detectors weren't very good. So we've worked on detectors, and we found these 2 Italian guys, Renato and Nicola. They were working at the Science and Technology, STFC, Rutherford Appleton Lab at Harwell. And Wazi Faruqi, the one on the left, he came from Pakistan originally and did a PhD at Imperial College in London. He was recruited by Hugh Huxley to work on muscle at LMB, and then Huxley went to Brandeis, and Wazi said, okay, I can do better detectors for X-ray crystallographers, but they didn't want it. So Wazi came and worked in our group from 1990 onwards, doing development of electron detectors. So originally it was phosphor fiber optic, and then Wazi met Renato, who did his PhD in 1996, and they were doing very, very big CMOS detectors, very slow, very big. But 2002, we started to work. So Wazi, Greg, Renato, Nicola started to work on how to make the CMOS detectors better for electron microscopy. And that took till about 2002, 2012. And then one of these 3 detectors, that was our project with Renato and Nicola, that was the most important factor in getting over the final hurdle. But there were lots of little hurdles before that. And so now, Nicola is building the next generation of detectors for these cheaper microscopes. still at the Rutherford Atom Lab in Harwell, but Renato has now gone to Barcelona and started his own company. So although we used to collaborate with them as a team, we now collaborate with both of them separately. So we're somewhat promiscuous in our detector collaborations, but both of them are doing, and the next generation of detectors won't be, it'll be another year or two into the future. So yeah, so I suppose you could say, so my role has been to help diagnose the problems and then persuade other people to solve them. And then once they've solved them, we buy in the technology. But I think this is exactly what happens in most fields, actually. You've got some people trying to make it better and other people telling them why it's not good enough. And then eventually, if it's a lot of money, you can't really do proper development without a company. So you have to have some skill and knowledge in how to do translation from lab-based prototypes, which are usually a bit clunky and they go wrong and it takes weeks and so on. And then once it's gone through the commercialization production process, which they have to make hundreds, they have to be foolproof and they have to have service engineers, and it can't depend on one person. So the academics can often rely on insights and expertise from one or a small group, but eventually it has to go worldwide. And then actually you want competition as well. Otherwise, if one company has monopoly, they do it really well and the price goes really well. So to keep the price, so we've currently got 2 companies that we're encouraging to make these inexpensive microscopes on the grounds that if you had one, they would take it and run. So, and I think a lot of the people here will have experience of how do you deal with the lack of, how do you prevent monopoly developing, but still do it in a way that's fair and allows them to make profits that are good enough that they don't say, I'm not going to do that, it's just not worth our time. So there's some middle line you have to do.
Richard, thank you. Sorry, Richard, thank you so much for such a wonderful and beautifully presented, and just the talk, and the images themselves are just incredible, but also to hear your intellectual biography. Unfortunately, we need to bring this session to a close. Also, it's incredibly generous for Richard to be here because he's actually jetting off to to Germany tonight to do an experiment tomorrow morning. So it's, which makes it even a rarer pleasure and privilege to have you here today. So please join me in thanking Richard Henderson again.
Thank you very much, David. Good. Definitely, I didn't talk tomorrow.